Okay, I'm going to say this on the recording so that you will hear it and other people will hear it. Oh, there's slim pickings today. There's not a whole bunch of people missing. Registration for fall starts Monday for DRC students and the next Monday for the rest of you. So if you want classes, Yeah, for fall. Yep. And, If DRC is Disability Resource Center, disability students. And then they stagger it, right, based on how many hours you have. 45 hours gets to register first, and then I think 25 hours, and then anybody new. So that starts the 8th for most of you, right? So Monday after next. FYI, biology classes will fill up instantly. My classes will be full within about 2 hours of opening. So not that you necessarily want me, but if you want somebody whose classes fill really quickly, you need to be on there, usually at midnight, because don't they open at midnight? Yeah, they open at midnight.
What about your first class? Is that okay?
Oh, it'll fill. Yeah, it's usually the first one to fill. Yeah. So I just wanted you guys to know, because I like you more than other people. Other people, I haven't met them yet. I don't even know if I like them. Maybe I don't. Okay. We're going to finish the heart. Even if we don't finish it, you're going to be tested on it. So we probably will almost. We almost finished it in the last class, but I think we'll probably finish it in this one. Okay. So let's talk about the basics. Now, how many of you guys haven't been to lab yet? Okay. So it's not going to be that hard if you haven't been to lab. If you did it in lab, this will reinforce it. If not, we'll introduce it to you and you'll actually get to see it in lab. I do want to reiterate what I said last time. When you go to lab this week, make sure that you are either involved in the dissection or get to see it afterward and put your hands in it, right? Feel the textures, stick your fingers through the valves, show yourself viscerally the way that they work, because it's harder to forget it when you actually see it that way. Okay, and make sure that you do a fresh heart too. You get to actually see a fresh heart and not just a preserved one. So let's talk about how the heart actually sits within the thoracic cavity. And by the way, when you look at the heart, don't just look at the isolated specimens, go look at the cadavers, because you can't really see how it sits with reference to the lungs. And you can't see the pericardial cavity unless you actually go look at the cadaver. And some of the cadavers, the pericardial cavity is completely gone from the front, but if you lift up the heart, you'll be able to see it around the back, right? You'll be able to see the sac and where the cavity was. So get in there, look around a little bit, right? Okay, so here's how the heart sits. It's kind of counterintuitive. It's not like the box that I showed you, of course, right? Don't look a damn thing like the box, but it gets you started. But it doesn't, it sits just behind the sternum and the ribs, but not centrally located, and it's not symmetrical. So it's actually asymmetrical two ways. First off, the tip of the heart, which is called the apex, sits more to the left side, so the heart is more left than right, okay? And then second, so it's asymmetrical that way. Second, it's twisted, so that from an anterior view, you're going to see more of the right heart than the left heart, which makes it look like the right heart is bigger, but it's not. They're almost the same size on the two sides, right? So the heart sits like this and like that. That makes sense? So more of the heart is to the left side than to the right side, which is why, historically speaking, when you did like the Pledge of Allegiance or whatever the hell, you did that. So, and it's of course sitting in the mediastinum. Somebody remind me what the mediastinum is. Yeah, it's a space between the two pleural cavities, right? It's not a cavity itself simply because it doesn't have its own serous membrane, right? What's running through the mediastinum would be like your trachea, your superior and inferior vena cava, your aorta, right? And also the heart within its pericardial cavity. We'll talk about the cavity in just a second. Does that sound familiar? Okay. All right. So now, okay. So when your doctor, nurse, whatever, wants to hear your heart beating with the stethoscope, what they're going to do is they're going to rummage around, and if you got a boob, they got to move it up, right? Especially if you're over 30, right? Probably was up here at one point, but okay. So, so you have a couple of kids, and you're like, where is it? They are going to feel between ribs 5 and 6, and you can actually feel that. You can actually like find the clavicle and then count down with the ribs. So right below the left nipple, especially if you don't have breasts, right, between ribs 5 and 6 is where the heart comes into contact with the anterior thoracic wall, and it's easiest to hear it, right? You can hear it from other perspectives, but that's where it's actually loudest. And that's usually where the doctor will actually look around for it first. So the pointed, although this is counterintuitive, the pointed inferior end of the heart, the point right here, it's called the apex. Apex means top, but it also means point, right? The apex, and then the flat top portion is called the base. So think of it like as an inverted mountain. Does that make sense? So here's the base. Here's the apex. The base is going to be important because all the big vessels come and go near the base, right? And then the apex is actually the bottom portion. Okay, so the heart is surrounded by the pericardial cavity. And we've learned about the pericardial cavity before, but we need to add a little bit more detail. Okay, so when we look at the pericardial cavity here, what I want you to understand is if the pericardial sac is intact, you can't actually see the structures of the heart, kind of like the dura mater around the brain, right? The dura mater is still there. It's opaque enough that you can't really see much. So this one actually has most of the pericardium cut off. So the pericardial cavity, if you look at it, so we already know some things about it. We already know that the heart sort of develops and then pushes into like a softly blown up balloon, and you end up with a layer of serous membrane and then a space and then another layer of serous membrane. Sound familiar? Okay. So that's what you're seeing right here. If you take a little chunk right here, you can see the parietal pericardium, right? Then a space. What's going to be in that space? Serous fluid, right? And then the visceral pericardium. They're just making it extra wordy. I don't know why the hell they did that. It's not necessarily parietal layer of the serous pericardium. Parietal pericardium is fine. And then this is visceral pericardium. There's another commonly used name for the visceral pericardium, which is the epicardium. And of course, there is going to be a small cavity called the pericardial cavity between them, just containing serous fluid. But what I didn't teach you guys before, because it's actually not a serous membrane, is on the outside of the parietal pericardium, you have a thicker connective tissue layer that is actually more structurally stable and more protective. So superficial to the parietal pericardium, you have a layer of dense irregular connective tissue called the fibrous pericardium. That's the opaque sac, right? And it's dense irregular, so it's not going to be stretchy, right? And it's actually going to be pretty protective. And when you see it around the cadaver, it's opaque. You can't see any of the structures of the heart unless you like slice through it. So this is the fibrous pericardium that is superficial to the parietal pericardium. That's what it's superficial to. So if we start to move in, we can move in and see the wall of the heart itself. So epicardium first. So if we see the epicardium, which is the same as the visceral pericardium, okay, and we move in, we're going to run into this layer, which is cardiac muscle. They call that the myocardium, which just means muscle heart, right? And then the innermost layer is going to be called the endocardium. So let's look at that in a little bit more detail. So if you take a chunk of the heart wall from anywhere, it's going to be really similar to this. The only difference will be how thick the myocardium is, really, okay? It is going to be thinnest in the atria, yeah? Thick in the ventricles, but thickest actually in the left ventricle for reasons we'll see today. The left ventricle has the thickest. But regardless, what it's going to have is the superficial surface of the heart itself is going to have visceral pericardium, also called epicardium on it. And do you guys remember that serous membranes are made of, with loose areolar connective tissue deep to it, right? And then greater and lesser amounts of fat in the areolar connective tissue. So you do have fat on the surface of your heart. You start building up fat on the surface of your heart when you're a teenager. How much fat you have on the surface of your heart has to do with your genetics and also has to do with your lifestyle choices. All of us are going to have fat on the surface of our heart, but if you have a lot of animal fat in your diet, it starts to build up on the surface of your heart, and for reasons that we don't understand, or at least I've never actually read an argument that I buy about it, the organ fat is worse for you than subcutaneous fat. the fat that's actually on the surface of the heart. And all of us have some of it, but when we, for instance, get a cadaver who's really obese, and I try super hard not to get those because they're so hard to dissect, it's like hours and hours and hours before you even get to the muscles. When we get a cadaver that's super obese, there's so much fat on the surface of their organs. And for whatever reason, that correlates directly to really poor cardiovascular health. And there's a genetic predisposition to that too. Like you can actually be thin and have a lot of organ fat, and that's just related to genetics. But regardless, so we're going to move in, and then you are going to, so this is epicardium, this is the myocardium, which is just what kind of tissue? Cardiac muscle tissue with a whole bunch of capillaries in it. So it's really, really vascular, okay? And then the inner layer is called endocardium. And really, it is a kind of serous membrane-ish stuff, but it's not on the outside, it's on the inside. Its primary job is to be super duper slippery. Any idea why it's going to be super important for both the inner surfaces of your heart and the inner surfaces of your vessels to be as slippery as you can get them? Yes, because if you catch platelets, right? Platelets, regardless of whether there's a hole, when they congregate and they don't move along, right, they start to attract other platelets. And when they start to do that, they start to form a clot, whether there was a hole or not. Okay? So I always like think of platelets like teenagers. They need to not congregate without supervision. Some's going to catch on fire or somebody's going to get pregnant. That's what's going to happen, right? It's going to send out a group text, right? Drink all your damn booze, right? Somebody's going to get knocked up. Maybe that was just Texas teenagers, but I really don't think it's that different here. You guys have a really, really high rate of teen pregnancy. It's just that then you get married. Still teen pregnancy. Okay, so that is the layers that we're talking about here. Now remember that we already talked about pericarditis way back the beginning of the semester. Does that sound vaguely familiar? And pericarditis is simply when one of these serous membranes gets irritated and you start to overproduce fluid to compensate for that. And since the bag, the outer portion of the bag is made of dense irregular connective tissue and it's not stretchy, pericarditis puts pressure on the outside of the heart that reduces filling and reduces pumping, right? And so you can die associated with pericarditis because the bag doesn't stretch. It just squishes what's going to happen. Okay, so we did the heart wall layers, and now let's start going into the actual anatomy of the heart. Okay, so of course the heart doesn't look like my drawing on the board because we all know that my drawing skills are limited. Okay, but it looks kind of like this, only it's not color-coded. Don't you wish it was? But it is not color-coded. This is a preserved heart. The actual fresh pig heart that you're getting looks very similar to what a fresh human heart would look like. And it looks kind of like this. This is a GIF of, it's pretty cool, right? GIF or GIF? GIF. See, I think it's GIF, but I sound weird when I say it. So this is an actual pig heart. The only reason that you can tell it's not human is because it's got these two branches of the superior vena cava. So that's kind of what it looks like when it's fresh. So I think it is GIF. Somebody who knows always corrects me on it, and I just go back to the other way. Okay, so what you see is two atria, two ventricles, but remember I told you it was twisted. So this is actually the boundary between the two ventricles right there. So you're seeing a hell of a lot more of the right side from an anterior view than the left side, but don't assume that the left side is smaller, because if you flip it around backwards, you see a hell of a lot more of the left side from a posterior view than the right side, right? So it's like twisted in the way that it sits in the body. So the two upper chambers are, of course, called atria. They are not very muscular, especially when you actually see the fresh specimen. That's why I want you guys to look at the fresh specimen, because when you look at the preserved specimen, it makes it seem as if the walls are pretty hardy, but when you use a fresh specimen, they're just flappy, right? There's not even much to them. They kind of feel like skin, right? So these two are primarily receiving chambers, right? they are primarily receiving blood. They do contract, but their contraction is really just to push 20% of the blood from the top to the bottom, right? They don't really do much pressure generation themselves with contraction. So these are receiving chambers, and these are your pumping chambers. So these are the ventricles. So atria, ventricles. So let's just look at the atria and the space between them real quick. So the space between the atria, or actually the partition, I should say, between the atria is named logically. And most of the stuff here is named logically. It's called the interatrial septum, which, what is a septum? Divider or partition or separation, right? So the interatrial septum, how would you find it? Stick your fingers in each of the atria and find it. It's really thin. It's not very big, okay? The interatrial septum allows for complete separation of the two sides of the atria because, of course, in an adult, the two halves of the atria are going to carry different kinds of blood, right? One oxygenated, one deoxygenated. Everybody good? Okay, so here's the interatrial septum. In the wall of the interatrial septum, if you are good at looking for it, it's kind of hard to find. You can find a little depression in an adult called the Fossa Ovalis. What that is the remnant of a fetal structure, because we're going to do fetal circulation in detail in the next set of notes, but let me just give you a preview. When you are little Cletus the fetus in your mama's belly, your lungs aren't doing anything unique. Yes? They're just growing like other tissues. They aren't actually getting oxygen from the air and delivering it to you. So do you really need for your heart during fetal development to function as two pumps, or can you use it as one? You just use it as one, right? Because the mom is delivering all of the oxygen into your bloodstream. Okay, so in order to allow you to pump as one, you just leave a hole open between the two halves. When it's open, it's called the foramen ovale. When it closes, it's called the fossa ovalis. In most of us, it will close with your first breath, but it takes a while for it to knit, but the pressure actually closes it with your first breath. And when it closes with your first breath, you get a good separation of oxygenated and deoxygenated blood, right? And your O2 saturation goes up. It'll go up again when something else closes. But you can still find the remnants of that in an adult called the fossa ovalis. Every once in a while, it doesn't close in people. And then you have a hole in your heart, right? And what you can imagine is if you have a hole in your heart and you're having mixing of oxygenated and deoxygenated blood, your heart's going to have to work harder to compensate for that. Does that make sense? Okay. So if it's bad enough, they'll go in and surgically close it. But if it's something you can manage and the surgery is riskier than leaving it open, then they'll just leave it open. And a lot of times with kids, it will close itself during the whole development process, especially from babies. Okay. So the external landmark that separates the atria from the ventricles goes all the way around the heart, and it's called the coronary sulcus. It's that depression between the atria and the ventricles. You got a whole bunch of blood vessels running in it. And it runs around the anterior side and the posterior side. So coronary sulcus runs all the way around. Does that make sense? Okay. So not that it's hard to find the difference between the atria and the ventricles because they're super easy to see. They've got totally different textures. Okay, and now we are going to move into the ventricles. When you move into the ventricles, the internal division between the left and the right ventricle is called the interventricular septum. Again, that should make sense. It's thicker, right, than the interatrial septum. It's pretty muscular. In most people, that's completely intact. Every once in a while, somebody actually still has a hole in that one, even though holes during development are not as common in the interventricular septum. They actually do exist. So here is the interventricular septum. Now, it's easy to see when you're on left versus right side with a coronal section of the heart, but how do you tell where you are if the heart is intact? This landmark right here with all of the blood vessels in it, right there, that actually marks the difference between the left and the right ventricles, and that's called the interventricular sulcus, or the anterior, I should say, anterior interventricular sulcus. So break that down. Anterior is obvious. Interventricular is between them, and it's a little depression. It doesn't look like a depression because it's usually got fat and vessels in it, but you can find it because it's always got those vessels running through it, right? Anterior interventricular sulcus. That's how you tell from the anterior side that everything to this side of it is left, right? Everything to this side of it is left, and everything to this side of it is right. Good? How about the back side? How can you tell left from right on the back side? Well, there's a sulcus back there as well. So this guy is sitting on top. Each of these are. This is sitting on top of the interventricular septum, right? This is sitting on top of the interventricular septum on the back side. The back one is called the posterior interventricular sulcus. It has blood vessels running in it as well. We'll learn the vessels a little later, okay? So everything over here is left, yeah? And everything over there is right. So if you know what you're looking for, you can tell left from right even on an intact heart. Good? Okay. All right, so those are your landmarks. Okay, so now let's get into the specific chambers. Okay, we're going to start on the right side of the heart. It's arbitrary where you start. The right and the left have a lot of similarities. We'll look at the differences as well. When you get into the right heart, here's the right atrium. See this little flappy portion right here? It looks like a puppy dog ear. That's called the right auricle. You also have a left one. It's really just this flappy portion that when your heart gets really full, like when you're exercising, you can put more blood into the atria. It can actually open up and make room for more blood for more venous return. So that's what the auricle is. You've got a right one and a left one. Then if you look inside the right atrium, how can you tell that you're in the right atrium? The way that I tell is the two big honking vessels that dump into it are right on top of one another. You have the superior vena cava and the inferior vena cava, and they are coming into the right atrium, right? So those two big honkers right there that are right on top of one another are the superior and inferior vena cava. So if you were looking at a specimen and it's got two big vessels, or sometimes the vessels get cut off simply because I try to tell the butchers, will you please leave the superior and inferior vena cava attached? They don't know what the hell I'm talking about. They got a whole different area of expertise, right, than me, right? So, but if it's got two big holes that are on top of one another versus four little holes, because the left atrium has four little holes that are lateral to one another. Does that make sense? So you can tell where you are based on that. And that is one of the reasons I think it's super useful for you to stick your fingers in things when you're looking at the heart, right? Figure out where you are by sticking your fingers in things. Okay, so the right atrium is going to have these two big openings into it, the superior vena cava and the inferior vena cava. Let's put this together with stuff that we learned last time. If we're in the right heart, what kind of blood are we dealing with? Deoxygenated blood. Which direction is blood going to be going through this vessel? Down, because it's delivering to the atrium, right? Which direction is blood going to be going through this vessel? Up, because it's delivering to the atrium. These are delivering the vast majority of the blood back from where? Lungs or body? Body, yes? And specifically, what the superior vena cava primarily does is drains the head and the arms into the heart, yeah? What the inferior vena cava primarily does is drain almost everything else into the heart, right? So the blood is returning back to an atria. And this correlates to what we talked about last time. Atria are receiving chambers. That means that these are what kind of vessels? Arteries or veins? Yeah, they're veins. Yes, good. Even though they're huge, right? We're used to thinking of huge vessels as arteries, but they're not. And interestingly, when you look at them, you will see that they collapse too. The aorta really stands up and the pulmonary trunk really stands up, but the superior and inferior vena cava are just flat on a cadaver. I mean, they're not flat on you, but okay. So you're going to dump into here. There's one other vessel that dumps into here called the coronary sinus, but usually that opening is kind of hard to find from inside the heart. We will figure out its significance a little later. So I'm in the right heart, specifically in the right atrium. The right atrium has on its walls this lumpy musculature called pectinate muscles. Pectinate muscles. You've got pectinate muscles on the left side too, but they're a little harder to find because they're mostly in the auricle. So notice lumpy musculature here and also lumpy musculature down here. Does anybody know why the walls of the heart might want to be lumpy instead of smooth? Any idea? It doesn't have to do with surface area. It took me ages to understand that. I thought it was the coolest thing once I finally understood this. Okay, so what the heart is going to do is it's going to squish, right? And when you squish and you have wet surfaces, if you have flat wet surfaces, you get stuck together like two microscope slides. Yes? Via hydrogen bonding. But if you have lumpy surfaces, what happens is you don't get stuck together, right? So the lumpy musculature on the walls of your chambers keeps the hydrogen bonding from sticking the walls together, which is kind of interesting to me. So you've got pectinate muscles here. You have them on this side too, but they're kind of harder to find. And especially since the left atrium is kind of smashed back behind vessels, right? And it's a little harder to see anyway, but you do have it there as well. So now I'm going to move from the right atrium through this valve, which is the right atrioventricular or tricuspid valve, to the right ventricle, okay? So I want to talk about valve function and the logic of valves so that you guys really get it, because people really, really misunderstand it even after they've had anatomy and are in physiology. So first thing, the valve is not muscle, it's just connective tissue, and it's really slippery connective tissue lined with endocardium, okay? So this one is called tricuspid because it has three big cusps or flaps, not just three because there's more, but three big ones versus this one that has two big ones. Why 3 versus 2? Evolution. It just evolved that way. Whatever our ancestor was had 3 versus 2. It doesn't seem to have any particular significance, and one is not particularly stronger than the other either. Okay, just like an evolutionary anomaly. So now let's pay attention and figure out how this valve functions. So here's what people quite often assume by looking at this. They assume that these muscles, called papillary muscles, are responsible for pulling the valve open because they look kind of like they would do that. But that's not how valves function. So here is how valves function, both of these, but we'll talk about them simultaneously. What causes those atrioventricular valves, left and right, bicuspid and tricuspid, to open is an increase in pressure on the top of the valve, because the valve acts kind of like a swinging door. Why does a swinging door open? Well, when you push on that part of it, it opens, right? So here's how the valve opens, or why it opens. Blood is continually returning to the heart through, for instance, the superior and inferior vena cava, right? And when it returns to the heart, the pressure builds up in the atrium, yes? Because it's full, right? And when the pressure builds up in the atrium so that it's distinctly larger than the pressure in the empty ventricle, it will cause that valve to open, yeah? Pushing on the back of a swinging door causes the valve to open. The pushing is blood. Blood's pushing, okay? So that causes the thing to open, right? And then blood moves from the atrium to the ventricle, yeah? So let's look at the ventricle in detail. So valve, a little bit more detail about the valves. So this is called the tricuspid valve or the right atrioventricular valve. It is attached to this lumpy musculature on the wall. Those specific little projections of lumpy musculature are called papillary muscles because somebody thought they looked like a nipple. I have to name every damn thing after a nipple if you possibly can when you are a Catholic monk doing anatomical research in the 1500s. And you're like, I'm pretty sure that's what a nipple looks like. Well, it kind of does when you look at this. Look at this cool picture. That's a papillary muscle attached to the heartstrings called chordae tendineae. So I've got the flaps here, right? These are called chordae tendineae, and they attach to this papillary muscle, which is right here. You can see the papillary. Here's another cool picture of the papillary muscles. You can actually see this is looking down from the atrium into a ventricle, so that's pretty damn cool. What people assume by looking at this is that the papillary muscles pull it open, but that's not the job of the papillary muscles. Go ahead.
Just going back a little bit, can you go over that mnemonic try to do right?
I haven't talked about it yet. I'm about to. Okay? So, what's going to happen, so let's talk about how you close them. So we know they open due to a pressure differential. Now I want to talk about why they close. Okay? So think about the tricuspid and the bicuspid simultaneously. How do you remember tri versus bi? That's what he was just talking about. The way I remember it is try to do right and just buy what's left. Right? So you can remember that, right? Try to do right. Okay? Because we're all trying, at least at certain moments. Maybe not at every moment, right? But at least at some moments you're trying to do right and just buy what's left. Right? So tricuspid on the right, bicuspid on the left. Okay? All right. So I want to talk about why those things are going to close. Okay? Why they are going to close is that now I just put a whole bunch of blood in the ventricles, right? And then the ventricles are now full and they are going to start to contract in the walls, yes? And as they start to contract in the walls, what I'm going to do is push on the back side of my swinging doors, yes? And as I push on the back side of my swinging doors, it will close them. But why don't they swing backwards? Because the papillary muscles, you just told them, red rover, there's blood coming over, and they get ready, right? And they're holding on to the chordae tendineae. So the job of the papillary muscles and the chordae tendineae is not to open them, but to keep them from swinging past closed. Does that make sense? Okay.
So the blood pushes.
So you're contracting the ventricles, and the way the ventricles contract is bottom and sides up, right? And as they do that, that's going to be important for pushing it out into the arteries, but we'll get to that in a second. So as you push, right, you're going to push on the back sides of those valves, right, as you push bottom and sides up, and you're going to push them closed. And if you have healthy papillary muscles and chordae tendineae, they're going to go closed and not flat backwards, right? Does that make sense? That's how the opening and closing of those valves. It's really just due to pressure differentials, isn't it? One side versus the other, okay? So now let's get into the ventricles, okay? So the ventricles are, of course, thicker musculature than the atria because they are pumping chambers, whereas the atria are primarily receiving chambers, okay? When they contract, they're going to increase the pressure in the ventricles. Let's look at a couple of things on the walls. Of course, we have these papillary muscles that are sticking off, but we also have this lumpy musculature that's all over the wall of the ventricles. That lumpy musculature on the wall of the ventricles is called trabeculae. Remember learning trabeculae before? They were the little beams in spongy bone, yeah, in spongy bone. Trabeculae carnii, which literally means meat beams. So you got some meat beams in your heart and you got some heart nipples. Meat beams and heart nipples, okay? So, so trabeculae carnii on the wall. So here's what's going to happen as you contract those ventricles. You contract the ventricles, right? When they're full, contract the ventricles. It will close the AV valves, right? But then you need slightly more pressure to open the next valves, which is the semilunar valves. Here's your semilunar valves. Let's pay attention to this one first. That semilunar valve opens at a slightly higher pressure than the AV valves close, which is good because you do not want both of those valves open at the same time, do you? No? Otherwise, blood would go backwards, right? So, contract the ventricles. AV valves close. Pause as the pressure increases enough to open semilunar valves. Yes? Okay, so the semilunar valve right here, they're called semilunar because the little cusps look like half moons. Why does that one open? Pressure in the ventricles is higher than pressure in the arteries, right? So you open it, right? And then blood leaves, okay? Blood is going to go out from the right heart to what organ? The lungs. So this is called the pulmonary semilunar valve. Pushes it into this vessel called the pulmonary trunk. The pulmonary trunk is what kind of vessel, artery or vein? Artery. Even though it's carrying deoxygenated blood, it's going away from the heart, so it's still an artery, yes? Okay, pulmonary trunk, which will split into the left and right pulmonary arteries and go to the left and right lungs. Good? Okay, so let's talk about why this valve closes. And that one's a little harder to understand. So I just squished blood out, right? And now what's going to happen is the ventricle's contracted and squished blood out and it leaves. And then the ventricle relaxes, and it's both relaxed and empty, so it's pretty low pressure, right?
The right one.
They're happening at the same time, but we're just going to talk about one ventricle at a time. Let's concentrate on the right one first, okay? So the right ventricle contracted, right? Pushed blood into the pulmonary trunk, opening the pulmonary semilunar valve, right? Okay. And now the right ventricle is going to relax. It's going to be both empty and relaxed, which means low pressure, yes? Empty and relaxed. As it is low pressure, what happens is the blood does what fluids do and try to go backwards from high pressure in the arteries to low pressure in the ventricles. Do you see that it would do that? Okay. And as it does that, it gets caught in the cusps and it will close the valve, right? So again, it's consistent. Why do valves open and close? Pressure differentials. That's why they do.
And how often is this happening? Sorry. Is happening?
Every single time your heart beats. Okay? So your heart is probably going 70-ish right now, but it can go up to 220. All right, go ahead.
Is there anything that's keeping those, the valve on the artery closed? Or not
Okay, so
closed?
this is a good question. Why do those not need chordae tendineae and papillary muscles? So it's a good natural question that occurs. Why don't they? And the answer is really that when the ventricles are contracting, they're pushing on a closed system. So the pressure gets pretty high. But when I push, think about this, when I push blood into the arteries, does most of the pressure stay there? No, it moves, right? Right? So most of the blood is going away. So you only need a slight pressure differential to close them. And unless somebody pinched my artery, it wouldn't be high enough to cause prolapse, right? Does that make sense? So because the blood is moving through the arteries, you don't usually have a prolapse problem in the semilunar valves. Is that okay? That makes sense? Okay. So does that answer your question? I think I got it. Yeah? Okay. So now we left. So where is the blood heading? It's heading to the lungs. We are going to go to the lungs. We are going to do gas exchange, right? So the blood that's coming back, what's the gas content going to be?
High oxygen.
High oxygen, low CO2. What chamber are you going to put it in? Left atrium, right? Receive into the left atrium. So it's going to go in here. It's hard to see how you dump it into the left atrium from an anterior view. So let me show you from a posterior view. From a posterior view, you can see the blood is going to come back into the left atrium from two pairs of vessels on most of us. Which direction is blood going to be going through there? Yeah, it's going to be going in, right? Okay, it's going to be going in. Are those arteries or veins? They're veins, yes, even though they're carrying oxygenated blood, because they are delivering back to the heart, they are veins. Their names are left and right pulmonary veins, because that's where they're coming from, right? Left and right pulmonary veins dumping into the left atrium. Everybody okay with that? Okay, dumping into the left atrium. So now let's go back into the left atrium. Most of it we've already learned. When you look at the left atrium, it looks tiny, but it's not. It's just tucked behind stuff, right? It's about the same size as the right atrium. It's just behind the pulmonary trunk and a little tucked back there. It's also kind of twisted. So you're into the left atrium. It's got most of the same structures. Usually it's hard to find the fossa ovalis from the left side, but it has to be in there, right? Because like it was between both sides. And the pectinate muscles are a little harder to find, mostly because they're in the auricle, but still pretty much the same stuff once you're in there. And then from the left side to left atrium to the left ventricle, this is my valve, which has three very commonly used names. Sorry. You can either call it the left atrioventricular valve, which is always safe, right? Because it's descriptive, right? You can call it the bicuspid valve because it's got two big flaps, bicuspid. Or clinically, it is most often, in my experience, called the mitral valve. But let me tell you why it's called the mitral valve. So do you see it right here, the two big flaps? Do you see something that looks kind of like this? Do you see that? Okay. Okay. So again, when we were first doing anatomical research, especially in Europe, you were either Catholic or you were not doing anatomical research. Those were your choices, right? So lots and lots of things were named for Catholic references. So, and this is one of them, because they said that that valve actually looks like a pope's or bishop's hat called a mitre. You seen the pope wearing this hat? So this is a pretty happy pope, right? That's a mitre, okay? That's why it's called the mitral valve, because it looks like an upside down pope or bishop hat. Their bishops get hats, guys. What the heck? No hats for you. You should be like, I need a hat, man. How are you going to know I'm special? Okay, that makes sense? So you're going through, you're on the left side. So now we go through this side. Now, please note the distinct difference in musculature between the left and the right side. This is not even hard to see on an actual specimen. Like, it's way thicker on the left side. So let me phrase this properly. Why does the left side need to be thicker musculature in the ventricle than the right side?
So it can pump to the body.
Has to pump to the whole body. Where does the right side pump? To your neighbor, right? Just to the lungs, right? But the left side has to pump to the whole body, all the way up to the head, which is against gravity, and then all the way down to the toes, even if you're Shaquille O'Neal. That's a long way, man, right? All the way down, right? So a lot more musculature in the wall of the left side than the right side. It gobbles up a whole bunch more oxygen. If it goes wrong, you're going to be a whole bunch more screwed really quickly. So the left side is actually pretty easy to tell when you're looking at the left side versus the right side. So on the wall, it has all the same stuff, not necessarily in the same proportions, but papillary muscles and then trabeculae, carneae. And when the left ventricle contracts, what it's going to do is push blood into this valve. That's the hardest one to see. I highly recommend sticking your finger in it to make sure that you prove to yourself where blood is going to end up when it goes through that valve. So when the left ventricle contracts, it's going to push blood to where?
The aorta.
Into the aorta, and the aorta is going to deliver blood to lungs or everywhere else. Everywhere else, okay? So you're going to push it into the aorta. It doesn't look like it's going into the aorta, right? So you push through that valve, and that's called the aortic semilunar valve, yeah? Semilunar, half moon, going into the aorta, and it pushes blood into the aorta, right? And the aorta, the portions are named for which direction blood is going through them. So you've got ascending aorta, aortic arch, and then descending aorta, and that's going to deliver blood to all of your systemic organs. Good? So that is how it works, and it opens and closes due to the same pattern that we noticed before. So what's going to happen in here? Let's just review the valves one more time. So the left ventricle contracts. Tell me which valve is going to open or close first when the left ventricle contracts. The bicuspid is going to do what? Close, yes? And why? Why is the bicuspid close? Pressure building up in the ventricles, yes? Okay, pause, generate more pressure, right? And then what valve is going to do what?
The aortic semilunar.
Aortic semilunar valve is going to open. Why? Pressure, exactly. I'm going to reinforce it until you get sick of it, right? And now, ventricle relaxes, and it's empty. What valve is going to do what? Semilunar valve is going to close, yes? Why? Pressure. Higher pressure in the arteries than in the ventricles. By the way, these pressure changes and valves closing are where your heart sounds come from. You can't hear valves opening, but you can hear valves closing and the blood slamming against them. So the heart sounds are lubbed up, lubbed up, lubbed up. So the lub is these two closing, lub. Okay, pause, and then you hear these two closing, dup. Right? And you're really hearing the blood slam against them as they close, right? Does it make sense that you would be more likely to hear that than them opening? So as the blood goes backwards and slams against the valve, that's what you're actually hearing. Lubbed up, lubbed up. And for these two are slightly more staccato, a little sharper when they close than these. Okay? That make sense? Okay. Questions?
So, yeah.
Go
Can
ahead.
you go back to that picture?
Mm-hmm.
Just making sure. So when the bicuspid valve closes, it's going to build up pressure, and then the aortic
Semilunar.
semilunar valve is going to close.
No, open. Open first.
And that's going to push the blood up into the aortic
Yes.
arch.
Yes, all the way into the whole aorta. Mm-hmm.
And why not into the pulmonary?
So these happen simultaneously. So we talked about left heart, we talked about right heart, separate than left heart. But FYI, here's what's really going on. It's just hard to teach it all at the same time. Blood is returning to both atria simultaneously. In most of us, what will happen is the blood returning to most atria simultaneously will cause the opening of the AV valve simultaneously. Yes? And then the left and right ventricles will start contracting simultaneously, and both AV valves will close simultaneously. and then pause, build up more pressure, and both semilunar valves will open simultaneously, blood will move out, and then they'll close simultaneously. So the left and the right heart in a healthy person work together. They don't do anything, they don't exchange between one another, but they work about simultaneously. Does that make sense? Okay, all right. Okay, so we got valve function, okay? And we understand why they open and close. Let's talk about what happens when they go bad, okay? What happens when they go bad is you can get what's called a murmur, and I have a heart murmur. My heart murmur and my other heart problem are related to the fact that I had rheumatic fever when I was a kid because my mom was a hippie and she didn't vaccinate us. So vaccinate your damn children, because there have been lots of problems associated with my cardiovascular system with that one. So here's what happens if the valves don't close properly or go past closed, right? So a murmur is anytime you can hear the blood going through a valve because it's almost closed, but it's really narrow. So it's really turbulent when it goes through a really narrow opening. Does that make sense to you? Okay? So it's going to be really turbulent and you can hear it. So that's called a murmur. The two big, most common causes for a murmur are prolapse, in which the valve goes to closed and then past closed. That's called prolapse, right? Okay. Or stenosis, in which your valve gets scarred or damaged, and so instead of being nice soft seals, like when you get a new car, right? You get a new car, it's really quiet, right? Especially if you get an expensive new car. Really quiet on the inside because the valves are all nice and soft. And then, if you're me, you make your damn car last forever, because I freaking hate spending money on cars. I wouldn't even own one if I didn't have to. So, and a whole bunch of hot summers and salty winters, right? And what happens to the seals of your car? They get all dried out, right? And then air goes through and your car gets noisy, right? Same thing happens if you actually scar your valves. They don't seal very well because they're not very pliable, right? Scar tissue, not very pliable. That's what happened with my heart because of rheumatic fever. So what happened is rheumatic fever likes to attack your heart, and it damaged my heart valves, right? And so they don't seal all the way. And so my cardiologist can hear the murmur. And because he's really good at it, he can hear which valve is murmuring. I can't even hear the damn murmur. I feel like a freaking idiot. Every single time he goes, listen to my heart, listen to your heart, listen to my heart, listen to your heart. And he keeps going, you hear it? You hear it? And finally I just lie because I don't hear it. And I feel like a dumbass. I'm like, I don't even know what you're talking about, but I trust you. So the blood is going backwards. Now, if your murmur is relatively minor, like mine is, they don't necessarily try to intervene because sometimes the intervention is more dangerous than the actual murmur. But if you have a decent amount of blood going backward, then your heart has to compensate for it by working harder, right? Does that make sense? So if it's not unidirectional flow of blood, then you're going to have to compensate for it by working harder. Mine's not major enough that they're going to do anything. They just listen to it, make sure it's not getting any worse over time. If it doesn't get any worse over time, they're just like, you just deal with that, right? I have some meds that I take that reduce the likelihood of the murmurs and the likelihood of the other problem. That makes sense? So you can either have a stenotic valve or a prolapsed valve. If it goes really far backwards, it can pop your chordae tendineae. And that happened with, you guys remember, well, some of you guys are so damn young, you weren't even alive. But there was a big, big settlement because of this diet drug called phentermine and fenfluramine. Each of the drugs was approved independently by the FDA, but then doctors figured out that they actually were a more effective diet drug used together. But the combination was never evaluated by the FDA. And one of the things that strangely happened with that is that it caused your chordae tendineae to calcify and then pop. And so, especially in this side, which is under the most pressure, so you had mitral valve prolapse, and you can have what my father-in-law has. He used fen, and he had mitral valve prolapse, and they had to go in and replace his valve with a mechanical valve. Sometimes they replace them with pig valves, though. So they can do either one, or cadaver valves if you can get a cadaver valve. So that's prolapse valve and a mitral valve prolapse. Okay, so the great vessels of the heart, we've already learned them. The great vessels of the heart are simply the big arteries and veins that are attached directly to the chambers. But let's conceptualize something. All of the great vessels of the right heart carry what kind of blood? Deoxygenated. So all these big blue bastards, those are associated with the right heart. Yes? Of course, it's not color-coded here, but it does help you remember which one's which. Okay? And then deoxygenated on the right side. Okay? All of the great vessels of the left heart carry what kind of blood? Oxygenated. So all of these red boogers right here, right? Those are all left heart vessels. Yes? Okay. And some people consider the coronary sinus a great vessel and some people don't. I don't really care. Mostly it's the big honkers that are associated, right? So those are called the great vessels of the heart and they go directly to or from the chambers. That's what the great vessels are. Everybody okay? Okay.
How do you differentiate when it's not color coordinated?
You just get used to looking at the specimens for a while, right? So you, after a little practice, right, you will be able to figure out, right, that here is, of course, the anterior interventricular sulcus. Everything over here is right. That means this big artery must be the pulmonary trunk, yeah? Behind it, because I know where to look for it after practice, right? This has got to be the aorta, right? And you know that the aorta is associated with the left heart and you know what kind of blood it carries. So this is stage 1 of studying, yes? And this is stage 2 of studying, right? But that's why they color-coded in the beginning. But after a while, it's not really very difficult. The heart makes a whole hell of a lot of sense. And honestly, after you get comfortable enough that you can take a specimen or a heart and find left versus right, then you're golden. It's kind of like with the spinal cord cross-section. As long as you could do anterior versus posterior, you're golden, right? Same thing with the heart. Can you tell when you're looking at the front versus the back, right? And it's not very hard to tell when you're looking at the front. Here's the front, 2 big vessels coming off of it. Here's the back, whole bunch of vessels coming into it, right? Tell front from back and left from right, and then you go from there, right? And it does take a little practice, but it's not as hard as you think. Have you been to lab yet?
Yeah.
Okay, cool. Okay, so those are the great vessels. All right, so now what we're going to do is talk about something that I misunderstood for an embarrassingly long time. Just a complete idiot. In retrospect, sometimes do you realize that you had a misunderstanding that literally everybody in the damn world understood but you? You're like, why didn't anybody tell me what a doofus I was? Well, here's one of my doofus moments that I literally didn't even understand until after I already had a degree in this crap. I don't even know what the hell my problem was. I think I'm just going to blame it on my redneck education. Okay, you ready? So the blood, the heart is a blood hog. The heart needs a disproportionate amount of blood and oxygen. So it's only about 1/200th of your body weight, but it requires about 1/20th of your blood and your oxygen. So it's really, really oxygen intensive and nutrient intensive, right? Okay. And it's really not very big. Like they always say the size of the closed fist, but not my fist. Like it's not this big. It's like a big dude fist. It's like a fist and a half, really. So it's not very big, right? Okay. But it's really, really energetically expensive. And here's the thing that I just did not understand for an embarrassingly long time. It's like, how on earth can the heart not get enough blood flow when the damn thing is always full of blood? Because Melanie did not grasp something very basic that you probably all get and can sit there and feel superior, that the blood that is going through the chambers does not deliver blood to the capillaries of the heart. So you can be completely full of blood and not be delivering any oxygen. Because we know, right, and now I know, that the only way to do gas and nutrient exchange is at capillary beds, right? So the blood that's going through these chambers is not hooked up to the capillaries from the inside. How do you get to the capillaries of the heart? You have to leave the heart and go from the outside in to get to the capillaries. So here's the thing that's really interesting and important to understand. The heart, and this is a cast of the heart showing you how many vessels it actually requires. In order to deliver blood to the myocardium, to the actual muscle of the heart, you are going to have to pump it out. Should the heart be a systemic or pulmonary organ? What kind of blood does it need? Oxygenated or deoxygenated? Yeah, it's a systemic organ that needs oxygenated blood, and everybody understands that, right? Okay, so in order to get blood to the heart, it has to leave the chambers of the heart and get pumped into the aorta, yes? Right? But the first two branches of the aorta give the heart blood, but from the outside, not from the chambers. Does that make sense? So the heart gets first dibs on oxygen and nutrients, but it doesn't get it from the blood that's running through the chambers. It gets it from the blood that's on the outside. Okay, so here's what we are going to do. And you have to do this for your test. So if you haven't been paying attention, pay attention now. Okay, this is your first concept of what we call a blood trace. Okay, so instead of just labeling the vessels, we're going to talk about how blood gets to and from different places in the heart. And this is your concept of a blood trace. So get ready. So I'll go through all the names of the vessels first, and then we'll do a couple different traces, okay? Okay, so when the ventricles contract, and here we're really going to be paying attention to the left ventricle, right? Because that's what's going to deliver oxygenated blood, right? Realizing that the right one's doing its thing too, but it's not relevant particularly here. So when the ventricles contract, specifically the left ventricle contracts, it's going to push it through what valve to leave? Aortic semilunar valve into this big vessel. What's that called? Yeah, the ascending aorta. And the only two branches that you will learn off of the ascending aorta are the left coronary artery and the right coronary artery. And those deliver blood to the heart muscle. Everybody okay with that? Okay. So which direction is blood going to be moving through this left coronary artery? Down, yes, right? From the aorta down onto the heart. And of course the ventricles need most of the blood. There are a couple little tiny branches that go to the atria, but you don't have to learn them, and they're impossible to find really unless you actually know what the hell you're doing. But just so you know, you are taking care of the atria, but the ventricles are the hogs really. Okay, so I've got the left coronary artery and the right coronary artery. They are hard to see, especially the left one, because the left one tucks out behind the aorta and then behind the pulmonary trunk and then under the left auricle. Right? So you can't really see where it's coming from, which is probably part of the reason that I didn't understand this freaking story, because I didn't see how it was hooked up. So it tucks out under the left coronary artery. So if you have an intact heart, the only place that you can actually identify the left coronary artery is right there under the auricle, because once it branches, it has a different name. So do you see? It actually is running all the way back there. So if you look at an intact heart, you really can only identify the left coronary artery right there, okay? And then the left coronary artery gives rise to two big arteries that you have to know. There's a bunch of small ones. You have to know all of the arteries in the heart. A, they're pretty variable between people, and B, they're kind of hard to find on an intact heart. So let's look. So the two that you need to know that are the branches of the left coronary artery are this super important one right here, called the anterior interventricular artery, or clinicians don't call it that. I don't know why the anatomists and the clinicians cannot get together, but most clinicians actually call it the LAD, the left anterior descending artery. Okay, you don't have to know that. I just want you to know it's the same one if you run into it in clinical situations. And this little bastard is super important because it delivers most of the blood to the anterior wall of the left ventricle. If it gets blocked, it'll kill you. So they call that one the widowmaker. Why? Because the left ventricle is pretty freaking important. Yes? So that one, which is the anterior interventricular artery that sits in the anterior interventricular sulcus, that we've already learned, okay? And then this guy will wrap around, do you see this one? Wrap around to the left side in the coronary sulcus. That's called the circumflex artery. And it's going to deliver most of the blood to the back side of the left ventricle. Good? Okay, circumflex. Okay, let's go through the other arteries first, and then we'll do the veins, and then we'll do a trace or two. Ready? Okay, so the rest of the heart gets blood from the right coronary artery. And so watch this for a second. The right coronary artery is easier to find because as it runs in the coronary sinus, it's still called the right coronary artery. Okay, so you could label it here, but you could also label it here, 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 all the way until it goes down, and then it's not the right coronary artery. Okay, so I've got the right coronary artery, which is going around in the coronary sulcus to the right side. Everybody with me? Okay, and it gives rise to only two arteries that you have to learn. There's more than two, and you can see them, but they're super variable between people. The two that you have to learn are this one, which runs at the margin of the heart on the right side, and it's either called the marginal artery or sometimes they say right marginal artery. I don't care if you say right because there isn't a left one, right? But it's on that side, right? Okay, so that's the marginal artery. And then this guy, all the way around the backside, going down the back. Do you see that? That's called the posterior interventricular artery that runs in the posterior interventricular sulcus. And it gives a whole bunch of blood flow to the back side of the ventricles. Everybody good? Okay, so here's what I just did. I just gave blood to all of those capillaries. Good job, right? And now I have to bring it all back up into veins and deliver it all back to what chamber? Right atrium. Does that make sense to you? It all has to go back to the right atrium. Go ahead.
Why is the blood to the atrium?
There's teeny tiny little arteries. They don't show them on this figure, but there are teeny tiny little arteries that come branch off of here, and they actually give blood to the atria from the underside, so they're really, really difficult to find, but they totally do exist. Of course, the atria, need blood too. You just don't have to learn them. I used to put them on the lab list, but nobody could ever find them, and they're not even on the models. So I just took them off. But obviously, conceptually, you need some blood in the atria too. Okay, so now let's get the blood back into what chamber again? Right atrium. Okay, so ultimately it's all going to end up back in the right atrium. You don't need to know a whole bunch of the veins that are involved in this, but you do need to know a few. And they are the great, middle, and small cardiac veins. Here's how they go if you watch. Watch. The great is on the anterior side, and then go back side before you come back to the front. The middle is back here, and then the small is over on this side. They are all going to dump into the same place, guys. The whole thing, the big vessel as it runs around, the coronary sulcus, yes, is called the coronary sinus. Sinus is just a wide vessel. It's not wide everywhere, but it's wide right there, okay? So let's talk about this one. Great cardiac vein. Which direction is blood going to go through the great cardiac vein? Up, yes? And it's going to dump into the coronary sinus, which dumps into which chamber? Right atrium, okay? This vessel right here, oh, by the way, the great cardiac vein runs with the anterior interventricular artery in the anterior interventricular sulcus, okay? This is the middle cardiac vein. It's on the back side, right? And the middle cardiac vein, which direction is blood going through it? Up, dumping into, not yet, coronary sinus, and then right atrium. Good, okay. And then this guy right here is called the small cardiac vein. And that runs either with or near the marginal artery. It's not sometimes necessarily right next to it, but it's close to it. That's the small cardiac vein. What does it dump into? Coronary sinus, and then into the right atrium. Good? Okay, so all of these are actually gathering the blood up. Okay, so let's look at the actual intact heart. Okay, so if you look at an intact heart, they actually run together, right? You can see this is the anterior interventricular sulcus, and it has an artery and a vein in it, right? And what's the artery? Anterior interventricular artery, and what's the vein? Great cardiac vein, which dumps into where? Coronary sinus, which dumps into the right atrium. Okay, so what I will have you do, and I'll go ahead and tell you verbatim, is that you are going to have to do a blood trace on your test on Tuesday to get blood from a chamber to a chamber, yes? And I want you to deliver blood to the anterior wall of the left ventricle, which is pretty damn important, and back. Can you do it? This is your first meeting of what we call a blood trace. And they're really not that hard. It's just a little logic story. So if I am going to deliver blood from a chamber to a chamber, and I'm going to go to the anterior wall of the left ventricle, which chamber do I start in? I start in the left ventricle, right? And then what valve do I go through? Okay, so I'm going to start in the left ventricle. I'm going to go through the aortic semilunar valve, right? And then I'm going to go into which portion of the aorta first, guys? Ascending aorta. And then from the ascending aorta, get me into the right vessel. Left.
Right, left pulmonary.
Nope, not pulmonary. Left coronary artery, which is sneaking back behind these. That's why it's hard to see, hard to figure out, right? Now, from the left coronary artery, get the blood to the anterior wall of the left ventricle. What is the only other branch I need to make? Anterior interventricular artery, yeah? Okay. All right. Did its job, goes to the capillaries. There's a whole bunch more arteries, FYI, but you don't have to know them all. Goes to the capillaries, drops off its oxygen, picks up its carbon dioxide, and now I have to get it back. Help me. Get it back. Great cardiac vein, which dumps into where, guys? Coronary sinus, which you can't see very well in that picture, but you can see pretty well in this picture. Coronary sinus, wrapping around, right? And then the coronary sinus will dump into what chamber? Right atrium. Good? Okay. So that's your first introduction to what we call blood traces. You will be doing boatloads of blood traces next week in lab. And you'll actually get to them in lab before we get to them in lecture because you've got a test on Tuesday. So that's why I think it's important for you guys to understand blood traces now, right? So this is a super simple blood trace. You guys have to do 6 different blood traces in your lab manual for next week, but this gets you introduced to the process. You can't ever skip capillaries, right? Because that's the point of all of it, right? And just start in the place that they tell you, go to the capillary bed, and then start going back via veins. Does that make sense? And generally speaking, you're going to start in a chamber and end in a chamber because that's literally how blood works, right? Go ahead.
As far as identifying these veins and arteries,
Uh-huh.
just remember, how do you do that? They're all kind of intertwined.
Oh, these guys?
Yeah.
Oh, on a specimen?
Uh-huh.
Okay. It's primarily by texture, but the lab instructors are usually more likely to ask you the arteries on a model. These arteries. Now, the big hunkers, you should be able to tell the pulmonary trunk from the aorta. But these little guys, it's so hard. And it's really hard to dissect them out. But FYI, this guy right here, the one that's standing up, that's an artery. Because the vein is collapsed behind it. So if it's like standing up and still relatively round, it's almost invariably an artery, okay? But with the exception of maybe Jill. Jill doesn't really use models. Almost everybody else will ask from the actual models when you're looking at the coronary arteries. Note also one little thing, and that is that they're coronary arteries, but they're cardiac veins. And you can't use those interchangeably. Coronary arteries, cardiac veins. Okay. Okay. So you're definitely going to have to do that. Okay. Everybody okay with that? Yeah? Okay. You're going to have to practice it. It just takes a minute, and then it's like immediately logical. And then you're like, I totally got this. It's not hard. Just get it there, get it back, right? I never ask you any artery that you didn't learn already, right? So you don't have to do any of the 25 other arteries that are on the heart, just the ones that you actually learned. So that will be basically a series of blanks in which I either tell you where to start, get you the capillaries, and then tell you where you end, or I intersperse some of the vessels, right? And you'd have to fill in the missing ones, okay? And it's going to be blanks. It's not going to be multiple choice, so you actually have to know them. Good? Okay. All right. So the last thing that we are going to talk about today is stuff that's kind of hard to see anatomically, but super important physiologically. And that is the innervation of the heart and the electrical activity of the heart. So let me introduce you to the innervation and electrical activity of the heart. Okay, so present in the wall of the heart, but not really very visible with gross anatomy. Histologically, you can tell them if you actually look at them, are these little blobs of electrically active cells called pacemaker cells. Okay? And I know you guys have heard of the pacemaker of the heart before, okay? And this is painted on the models, but when you're actually looking at a specimen, it doesn't really look very different with gross anatomy, but it does look different with histology. So I have this collection of electrically active cells. If there's a blob of them, it's called a node, but there might be a chain of them, and it's called a bundle, okay? What these are is cells that can spontaneously generate an electrical stimulus, spontaneously, and cause a contraction. Okay, so they're called pacemaker cells. One of the things that's important for you to understand about the heart is that the heart will contract without the nervous system telling it to. And that's totally different than skeletal muscle, right? Skeletal muscle doesn't do a damn thing unless you tell it to. But with cardiac muscle, it actually will. So let's meet these and figure out how this causes the series of contraction and everything that we talked about before. So let's meet the anatomy first. This node is called the sinoatrial node. It's the boss. You find it just inferior to the opening of the superior vena cava. Okay? That's the sinoatrial node. That's the one that commands the entire thing in a healthy person. At the bottom of the interatrial septum is the atrioventricular node, which is another node. It's like the underboss. It will do its own thing, but whatever command it gets from the SA node will override whatever it was already doing. And then we have a chain of these cells, and this chain of cells from the AV node goes down the interventricular septum. It's called the AV bundle, or sometimes the bundle of His from the guy who found it. So it's going to go down the interventricular septum, and then it's going to split and go left and right sides. This is the left bundle branch. This is the right bundle branch. Okay, these are going to take the electrical stimulus to the walls of the ventricles. But you only talk to the ventricles via the Purkinje fibers, which are the very things at the end. For those of you who have had or are having physiology, here's what's going to go on. Okay, you ready? The sinoatrial node is going to send out an electrical stimulus. For those phys people, it's sodium and calcium ions sent through gap junctions. Okay, and it will depolarize, right, the wall of the atrium. And then the cells will contract in the order that they got the stimulus. So they are going to contract in the atria, top down, top to bottom. Does that make sense? That's the order they're going to get the stimulus. And then when the atrium is almost finished contracting, the AV node is going to go, I got you, boss, and it's actually going to get the stimulus, but it's not going to talk to the ventricles until it gets to the Purkinje fibers. And this is really important for you guys to understand. The AV node sends a stimulus down the AV bundle, but doesn't talk to any cells and make them contract. And then it sends it up the sides and doesn't talk to any cells until it gets to the Purkinje fibers. The Purkinje fibers talk to the contractile cells. So how do the ventricles contract? Top down or bottom up? Bottom up. Yes? They contract bottom up. And this forces a lag time between atrial contraction and ventricular contraction. Why do you think the lag time might be important? Filling, right? You're going to fill, make sure you're all the way full, right? Before you actually contract, right? So then the ventricles are going to contract bottom and sides up. And please note that my papillary muscles also have Purkinje fibers, and they actually get the stimulus first. So you say, get ready, bro, red rover, there's blood coming over, and they go, I got it! And then the walls contract, right? And they're actually holding on to, so when the valve closes, it doesn't prolapse all the way back. Does that make sense? Okay, so this is called the conducting system of the heart, as in I'm going to conduct and tell you what to do when. Everybody good with that? So, and this works because the cardiac muscles have gap junctions, right? Because you don't need to tell every one of them what to do. You just need to get it started, and then it'll tell the neighbors, right? Good? Okay. So this is called the conducting system of the heart. SA node, AV bundle, left and right bundle branches, and Purkinje fibers is how that works. And this picture, by the way, the green is trying to show you the order in which things will contract. Start here, go here, right? Start here, right? And then go bottom and sides up. Good? Okay. So the last thing to talk about, as you guys are about ready to leave, is, well, if the heart does its own thing, why is it hooked up to the nervous system? Okay? So if you don't need the nervous system to tell the heart what to do because it spontaneously does it, what does the nervous system do to the heart? Parasympathetic and sympathetic, but they don't start and they don't stop the heart, which they do for skeletal muscle, right? Skeletal muscle, it won't do anything unless the nervous system tells it what to do. But here's the metaphor that works for me with the heart. I want you to imagine that the SA node is the starter on your car, and if the car isn't started, does it matter whether you hit the gas or the brakes? No difference whatsoever, right? That makes sense? So your heart has to be initiated and started with the SA node, but after it's going, you can change the rate. with the sympathetic and the parasympathetic nervous system. Does that make sense? So you do, of course, have autonomic innervation of the heart, but it has no capacity to stop or start the heart at all. We don't have any capacity, even clinically, to start the heart. We can stop it. I'll talk about that in just a second. So here is what we are going to do, okay? So this looks like a super complicated picture, but it's stuff that you know, okay? All right, so how do I slow down my heart? Well, the SA node is doing its thing, right? But what I can do to slow it down is send out a stimulus from the medulla oblongata, which we know controls heart rate, through this cranial nerve number 10. What is it? Vagus nerve, to the SA node and the AV node, and I can say chill, right? And it's basically like putting my brakes on, what the heart was already doing, okay? But the brakes won't make any difference if the car wasn't started, right? Or polar bear walks into the room, I can, from a different portion of my medulla oblongata, yes, say I need to increase the speed of cardiac muscle contraction. And this time I won't send it through a cranial nerve, I'll send it down to the thoracic region of my spinal cord, out to the sympathetic chain ganglia, to the SA node and the AV node, and it will tell it to speed up. Yes? So all your nervous system can do for the heart is increase or decrease the speed, but it can't start it. It has to be started with the SA node, which is why if you've ever seen anybody on TV or in any other situation do cardioversion in which they shock you, right? Are they trying to restart your heart? No, they are trying to stop it from misbehaving, and then they wait, don't they? They wait to see if the SA node remembers how to behave. And if it does, you are going to live. And if it doesn't, you are dying anyway. Okay? So let's talk before you guys leave about abnormalities of conduction, because one of these is the one that I have. So when you have fibrillation, what it means is they're not coordinated. Here's what should be happening with your heart. Atrial contraction, pause, ventricular contraction. Atrial contraction, pause, ventricular contraction. Yes? That's what should be going on. If they are discoordinated, usually because of some electrical abnormality in your heart, you can have, among other things, a fibrillation. This is atrial fibrillation, although I'm not coordinated to do it. Imagine that this keeps going on, so I'm going to fall over if I try, but the atria are going brrrr. That's the disorder that I have, the electrical disorder that I have, okay? But there's also ventricular fibrillation, and ventricular fibrillation is when the atria are doing their job just fine and the ventricles are going brrrr. Which one will kill you real quick if you don't stop it? Ventricular fibrillation, because those are the pumping chambers, right? It will kill you really quick if you don't, by a heart attack or a stroke, if you don't actually stop it, right? But atrial fibrillation, which is what I have, will not kill you very quickly most of the time. What it does do, though, is drop your blood flow by about 20%, because when the atria contract, it squishes the last 20% of blood from the atria to the ventricles. So it really, really makes you lightheaded, and sometimes I pass out when it occurs. So it squishes the blood from the atria to the ventricles, right? So if it's not doing that, then the atria are just, they're not getting the last of the blood out. So it really drops your blood flow, right? But it's not necessarily life-threatening, although it's potentially life-threatening. And it's primarily potentially life-threatening because that blood just sits in the atria, and when blood doesn't move along, it increases the likelihood of what? Clots, exactly. When I have a bout of atrial fibrillation that lasts for more than a couple seconds, I'm at increased risk of stroke and heart attack for weeks afterward. I have to shoot myself up with anticoagulants. And the whole time I was pregnant, I had to shoot myself up with anticoagulants 3 times a day because estrogen increases your risk of clots. That combined with atrial fibrillation was a double whammy. So the anticoagulants were necessary the whole entire time. Does that make sense? That's atrial fibrillation. That's how that works. So increases the risk of strokes and heart attacks and thrombosis and pulmonary embolisms. Okay, so we got done. Good. Okay, so I will see you guys on Tuesday. The test, again, covers the brain notes, okay, the spinal cord, spinal nerves, and autonomic nervous system notes, and heart, but no blood. I'll figure out what to do about blood. Either it'll be on your 4th test or we'll drop it. Not sure yet. But not on this test.
But the blood tracer.
Well, yeah, I'm talking about the fluid blood. Fluid blood, which is like erythrocytes and all that kind of stuff. Study brain. That's what people do poorly on. Study brain.